You are covered in bacteria, and I am as well. As a matter of fact, we're all covered in bacteria. According to a study called the Human Genome Project, there are approximately 11,174 different species of microbes living on and in us at one given time. This community of microbes that we're all just walking around with is known as your microbiome. Now, microbiologists study the microbiome. They also make probiotics, antibiotics, they study plant diseases, and many of them know how to make beer, cheese, and wine. So make sure you invite one to your next get-together. Even though there is a lot of diversity among the jobs of microbiologists, they all employ one common tool, the Petri dish. Upon first sight, the Petri dish looks pretty simple: lightweight, plastic, and empty. But what we put inside the dish is actually very complex. To begin, you first have to pick your media. Media comes in two forms: either broth, which is stored in test tubes, or in agar form, which is kind of the consistency of Jell-O once it dries. Once it dries, it provides a platform for the bacteria to grow in on the bottom of the plate. To do this, it's pretty simple. On the side of the container, it states 13 grams per 1 liter of water. So you simply fill your flask to 1 liter of water, and you measure out 13 grams of the media. Combining them together, you will then wind up with water on the bottom and powder on the top. We are going to want to take this and swirl it so it becomes in solution. Our next step is to cover it with tin foil and autoclave. Now, the autoclave is going to make our media sterile, and it does this by using high amounts of heat and high amounts of pressure. And it essentially boils the media inside the flask. And the cycle of the autoclave takes approximately 20 minutes. And when you open after the cycle, the glassware is going to be extremely hot. You won't be able to touch it. You have to use a rubber glove to remove it. Once you remove it, we want to set it on the bench top and allow it to go from boiling to room temperature. When we're waiting for that to happen, we can go ahead and sterilize our counter space. This is going to break the cell wall of any bacteria that might be living on the surface, and that way when we pour our plates, we don't contaminate them from the countertop. So, waiting for this to get warm, you may be wondering, why don't we pour it when it's hot? Well, if we did pour it while it was still boiling, we would accumulate quite a bit of condensation on the insides of the plates. And for future use, it provides a very wet environment, and the water tends to drip into your samples, and it's very undesirable. So now that we have a clean workspace, we line up the plates that we want to pour, we take off the tin foil, and we barely lift the lid, preventing any contamination from the air. So you kind of want to open it to like 1/4. You aim to pour about half of the plate in, and then we swirl it, and that's going to distribute the very dense agar to the rest of the plate. If we were to pour the whole bottom of the plates tend to be very thick, and you wind up wasting media. So once you finish pouring all the media from your flask into your plates, you then just allow them to dry. That takes about 1 hour, and then they're ready to use. I'll now open the floor to any questions, and after the questions, you feel free to come up and look at the finished products. Yes?
I have a question. I've seen a lot of that cell agar plate art online, and I was wondering if you've tried that.
I haven't tried it. It's actually a lot more difficult than you would think it would be. The way that it turns colors has to do with the particular bacteria that you put on it and how that bacteria reacts to the media. Sometimes bacteria turn colors and sometimes they don't. So a good background knowledge of how that all works, how that chemistry works, is pretty important. And quite honestly, I haven't had the extra time to sit and try to figure that out, but it's definitely something that I plan on doing in the future.
Very cool, thanks.
Anyone else?
Are there different types of agar?
There are many different types of agar. The difference between them is that basically the nutrients that each one has. So if you're trying to feed E. coli versus Staph aureus, you're not going to eat the same things. So you generally would probably put them on different types of media to select for those particular bacterias. However, this is nutrient agar, and it's basically like a Snickers bar for bacteria. Most bacteria will grow on nutrient agar, so it's a catch. This particular one is a catch-all for all bacteria.
Awesome.
Anyone else?
Thank you.
Thank you for your time.
Thank you.